Hello, my name is Professor Shinagi from Faculty of Pharmaceutical Sciences, Tokyo University of Science, Japan. It is my great pleasure to give a talk in this wonderful symposium, International Conference on Cancer Research, ICCR 2019. Today I'd like to show you some recent results on the development of new methods for detection, isolation, growth, and recollection of cancer cells in blood based on size discrimination of cancer cells and normal cells. As you know, one of the big problems of cancer is metastasis. And it is believed that metastasis is mediated by circulating tumor cells, CTC, that are cells coming out from primary tumor sites and go into the bloodstream and travel in the body and start the metastasize at the remote place. Besides, recent result suggests that CT cluster has more metastatic potential than monomic CTC. But CTC has a clinical value as biomarker for early cancer detection, diagnosis, prediction of prognosis, and monitoring therapeutics outcomes of cancer therapy. But the number of CTC is very small. Only 1 to 100 CTs are found in 1 billion red blood cells. Therefore, detection of and isolation of CTCs is very difficult. In this context, we have studied the program to develop the easy and convenient detections of CTC and for isolation, growth, and recollection of CTC. The following are the result of our interdisciplinary research in Division of Medicine Science Engineering Corporation Research Institute for Science and Technology, RIST, of our university. Especially, we are working on these researchers in a university and Tokyo University for the specific purposes, as I told you before. Okay, the first half about the easy and convenient detection of CTC using particle size analyzer and new PC softwares. We have focused on this instrument, particle size analyzer, just called flow cell, because this instrument has been designed to detect the micrometer size particles by using shape parameters such as circularity and aspect ratio, and shape parameters such as area diameter and inner diameter, and so on. Then we have, it was known that the cancer cell lines have larger size than normal cells, especially blood cells such as T cell, B cell, red blood cells. Then we have measured the size and the shape of these cancer cell lines such as JACAT, LLC, Herein, and B16 on this particle analyzer. And the result is shown here in this 2D plot with respect to the mean diameter and roundness of these particles. As you can see, cancer cells have over 10 micrometer size. On the other hand, this red blood has the diameter of these cells are less than 10 micrometer. Then let me show you how this machine works. It is now possible to take a picture. This is the blood sample including cancer cells, in this case SP2 melanoma cells, and found that this instrument, this software installed in this instrument detects the larger cells like this than blood cells, red blood cells. This software automatically detects the larger cells with marking in green like this. Remember that this is not the fluorescent, this is just a green color for the detection of abnormal cells included in blood. These results are summarized in the 2D plots like this with respect to the particle size and the number of the particles. In this case, red blood cells, T cells, B cells are distributed here less than 10 micrometer, and the cancer cells are very far in this area. By changing the plot by the volume rather than number, the sensitivity of cancer cells are much improved like this. Next, we have checked the size of LHC cells with these normal cells taken from blood or mice. And inner diameter of LHC was 12 micrometer. The inner diameter of other normal cells are less than 10 micrometer, as I told you before. The shape parameter of cancer cells, LHC cells, and normal cells such as platelets, red blood cells, white blood cells were compared in this slide. As you can see, there are almost no negligible difference between cancer cells and normal cells. But for the size of parameters are very different. As you can see, the size parameters of LHC such as inner diameter, mean diameter, length, and so on are much greater than those of normal cells, suggesting that the cancer cells are size parameters, good parameters for the discrimination of cancer cells from normal cells. Using this methodology, we have checked the size and shape of cancer cells in dog blood and cat blood. Using this is the normal dog and normal cat. This is the result of the analysis of blood from cancer-bearing dog and cancer-bearing cat. As you can see, there is almost no particles larger than 10 micrometer in normal dogs, normal cat blood. On the other hand, these large cells were detected in cancer-bearing dog and cancer-bearing cat. The number of these particles are 2 in 100,000, or only 1 in 41,000 particles. But sometimes white blood cells have large size as cancer cells, and the observation of these normal cells and cancer cells on particle size analysis suggested that cancer cells has an even surface like this. Also, the normal cells has smooth surface like this. This difference was quantified by using PC software as shown in this slide. I mean, we have drawn these blue line and red line passing through the center of gravity of these particles. And the brightness curve of this on this line and this line was measured and showing that the stationary point on this curve are very few, 1, 2, 3, 4, 1, 2, 3. On the other hand, stationary point on this line of cancer cells are more, 1, 2, 3, 4, 5, 6, 7, 8, 9, and 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Then we have calculated the sum of the stationary points along this line and this line, and it was found that normal cells has sum of the stationary points less than 10. On the other hand, the sum of the stationary points of cancer cells are over 10. Then using this knowledge, we have developed the new software for the detection of the cancer cells. This software was designed for Macintosh. Therefore, you have to start MATLAB first, this software first, in which the CCF Cancer Cell Finder, CCF software we have developed works. Then CCF was chosen and studied. In this case, we have chosen 4 examples. 1, 2, these are cancer cells, and these 2 are normal cells. And after starting this software, we are going to choose these 4 pictures, 4 files, 1, 2, 3, 4. After that, this CCF software suggests that these 1 and 2 have some of stationary point of over 10, like this. On the other hand, stationary point, some of stationary points of 3 and 4 are less than 10. Therefore, this software suggests that the cell number 1 and number 2 are cancer cells, and 3 and 4 are normal cells. Using this software, we have tested the real blood, like as shown in this slide, and CCF suggests that this one would be cancer cells and the other cells are not cancer cells. Okay, then we have succeeded in the development of easy and convenient detection of CTC, and the second half will be about the convenient methods for isolation, growth, and recollection of CTC using magnetic beads, microfluidics, glass beads filter. But my time is limited. I would have to show some result on the glass beads filter for this purpose. Then, as I told you, it is possible to detect the cancer cells on particle size analyzer, and there we found that there is a threshold between cancer cells and normal cells at 8 micrometer. To separate these size, we have decided to use glass beads filter like this. I mean, the glass beads were commercially available, and sintering these glass beads are having having the diameter of 60 to 100 micrometer. It is not possible to make the well-defined pore like this. And we assumed that the mixture, if you put the mixture of CTCs and red blood cells goes through this pore, but larger CTC are trapped on or in this glass beads filter. Besides, it would be possible to grow them after incubation in this filter. First, we have prepared the integrated glass beads filter, which we call iGBF, and we have collected blood from cancer-bearing mouse and trapped the cancer cells in this here and grown cancer cells in the 24 well disease. And after that, after incubation, GBF was put in the 5 ml tube and to collect the cancer cells from this CTC to the bottom of this tube by the centrifuge. After that, collected CTC cancer cells were analyzed on particle size analyzer and cancer cell finder software. Again, iGBF was used, and after the trap of cancer cells in this filter, the CTCs are grown in this 24 well disease and put in this centrifuge and collected by a centrifuge. Then in this experiment, the cancer cell B16 expressing GBF was injected to the mouse like this, and blood was collected after 2 weeks to trap the cancer cell like this. After centrifuge to remove the red blood cells, the CTC trapped in GBF were incubated for about 1 month. After that, these grown cells were collected and analyzed on particle size analyzer. The plot of this sample suggested that this contained the larger cells like this, and analysis of these 6 cells suggests that these cells have over 24, 25 micrometer size, and some of the stationary points of these cells are over 10, suggesting that these cells could be cancer cells. Moreover, it is possible to change size and shape of GBF at your will, and in this experiment we have prepared GBF having a diameter of 2 centimeters and used for the CTC trap in large amount of blood, 7 ml and 50 ml. After that, GBF was inserted to this folder and grown, CTC grown in these 6 well plates, and after that they collected using this 50 ml tube and analyzed by using these methodologies. Then let me show you how we use this glass beads filter. GBF is inserted between this apparatus instrument, and bovine blood containing cancer cells were put in this space. At the atmosphere pressure, red blood goes down through the GBF, and the CTC cancer cells are collected on or in this glass beads filter. It takes about 12 minutes to filtrate 7 ml of bovine blood. In this case, bovine blood contains heparin, but it is not diluted. Glass beads was inserted for use for the collection of cancer cells with in this apparatus, and after that, this filter is inserted to the GBF folder for the growth of CTC in 6-well plates like this. And after that, this folder is put into the centrifuge tube of 50 ml, and the CTC was collected by centrifuge to collect at the bottom of this tube. Let me show you how to use this GBF folder. GBF, after the filtration of cancer cells, inserted to the GBF folder like this, and CTCs are grown in this 6-well plate. After incubation, this folder is put in the centrifuge tube of 50 ml, and the CTC will be collected by the centrifuge in this tube. Besides, it is now possible to collect the CTC from large volume of blood. 50 ml of blood containing cancer cells are put in these bottles and circulated in this circulating tube by using this peristaltic pump, and blood is inserted to this folder, including GBF. In this case, GBF is included. This one, no GBF is included. As you can see, the red blood can be circulated like this if you put this outlet into this bottle. Therefore, it is now possible to collect the CTC from small amount of blood and large volume of the blood. In summary, we have succeeded in the development of easy and convenient detection of CTC and isolation, growth, and recollection of CTC. These results were reported in these 2 papers in Biological Pharmaceutical Bulletin and AC's Biomaterial Science and Engineering. And especially second paper was introduced in this website, Medicine Innovates. Therefore, if you are interested in this work, please refer to these 2 papers. In the final slide, I'd like to show you a picture of my research group. I am very proud of working with these capable persons, and I'd like to acknowledge the financial support from Japanese government and private foundation and our university. And finally, thank you for your attention. Thank you.